Hi, everybody. Thanks, Kevin. So I'll sort of pick up where Jerry left off, but I'll first provide sort of a little bit broader context about why we're doing what we're doing. So Jerry gave you some great examples of nitrogen response curves for corn yield, right? Some better behaved than others, but ideally we'd see something like this, right? You all are really familiar with that. Perhaps less familiar with this idea of a nitrogen leaching response curve, right? Which, in some sense, we expect to be kind of the opposite of this corn nitrogen response rate. This idea that as we sort of exceed the corn plant's capacity to take up nitrogen, we expect that nitrate leaching to increase disproportionately as the N fertilizer rate increases. And so really this highlights the importance of dialing in our nitrogen rate, right? We really want to optimize our yield and our economic return, but at the same time, we want to do this thinking about sort of the broader environmental context, right? Keeping our water underneath us fit to drink. And sort of the challenge, right, is that in many cases, the nitrogen rate that might actually be most protective for water quality, right, thinking about that 10 milligrams per liter, national standard, that may behave a little bit differently. And so we need to better understand the shape of this nitrogen leaching curve and how this interacts or does not, right, with our corn yield response curve. Okay, and so right, the challenge, right? There's gazillions of these yield response curves out there. The story for nitrate leaching is, there's a lot less data out there, and part of the challenge, it's really hard to do this. And I think one of the best data sets out there actually comes from colleagues in Iowa at Iowa State University, where they have some of these long-term drainage plot experiments. So essentially, they've got plots with individual tile drains where they're able to vary the nitrogen rate and look at the water that's coming out of that tile to get at, how the nitrogen management influences the nitrate leaching year over year. So this is a figure that shows you 16 years of those data from a corn bean rotation, right? You see in any particular year, there's a lot of variability, right? A lot of spread around any of these given nitrogen application rates. But if you have enough data, you can begin to tell a story, right? Consistent with that kind of nonlinear response of nitrate concentration to N application, right? And so really, one of the our tasks is to figure out how these shapes may or may not vary in Wisconsin cropping systems with differing nutrient management and with other conservation practices potentially as well. I think one of the challenges that we have is that unlike some of our land grant counterparts elsewhere in the Midwest, we don't have any of these established drainage plot experiments, and so that's one way we'd hopefully like to go in the future. But in the meantime, right, how else can we get a handle on this? Well, we're fortunate that, some folks in UW soil science 20, 30 years ago developed this really nice lysimeter approach that you may have heard something about. This is known as the equilibrium tension lysimeter. This is it's a fairly sophisticated way of capturing drainage water in a very physically realistic way. Essentially, we have boxes installed below soil where we're applying a fixed suction to that box that's really representative of the natural soil profile conditions so we can get fantastic data on drainage and nitrate concentration. Really, the challenge is that these are extremely expensive and labor intensive to actually do. It's difficult to envision how we could do this in the context of something like NOPP. Although, we've had, good luck with colleagues in Discovery Farms who have done this and a number of other places across the state. You may also be familiar with another common approach for measuring nitrate leaching. This is known as the suction cup lysimeter. Essentially, this is just like a tube that you stick in the soil, and on the end of it, there's typically a little porous suction cup. And so you can apply a vacuum to that tube, and you can suck some of the soil water in, and you can collect it in a bottle, right? This is potentially useful for getting sort of snapshots of nitrate concentrations in the soil water. But one of the challenges is that it doesn't really directly measure leaching, right? You're just getting a sense of the chemical characteristics of the soil water. It's also really labor intensive, right? You've got to go out frequently and apply the suction and measure the bottles. So I think useful, but maybe it's tricky to get at these questions of how nitrogen management really influences leaching. And so an approach that we've been using, is trying to sort of thread the needle. We call this the resin lysimeter. So this was developed more than 20 years ago by Susfolk and Johnson. It's been used by a number of other research groups around the Midwest. And it's essentially it's an open PVC pipe with a couple of segments here, and in the middle, this is a layer of ion exchange resin. And so this is sort of a substance that will bind onto nitrate and ammonium and other sort of charged particles in water that flows through the resin. So here's an example of what these look like. Sort of, one of the benefits, right? This is, relatively inexpensive. You can install them, and they sort of passively record the nitrate that leaches through. So you can have a lot of replicates. You can throw them out on a farm field, come back a year later and collect them. And so it's sort of a approach that's feasible to implement in something like NOPP. Want to highlight though some of the caveats, right? These are noisy data sets. Arguably, this is maybe giving us a relative value of nitrate leaching, not necessarily absolute. We know that when we install these little tubes in the soil, we're altering the soil physics to some extent. So we may not get the same kind of drainage through this lysimeter that we would under natural conditions. We can also have really high spatial variability, right? Because these are pretty small. But with those caveats accepted, right? If you get enough of these out there, maybe they'll tell you something useful. Okay, so here's a photo of the gang of folks installing some of these lysimeters up at the Chippewa site that Jerry was talking about. This is one of our ag water colleagues, Kelsey Highland, in a soil pit here that was excavated. She's carving a little pocket in the side of the pit, and we install one of those lysimeters in the side, fill it back in, right? So we have some undisturbed soil on top. Then we fill the pit back in. Come back a year later and dig these guys out and extract them. And so here's the data, right? And so this is the same experiment that Jerry talked about before, where we've got this whole spectrum of synthetic nitrogen rates with and without this liquid dairy manure. And so I'll walk you through this figure because there's a lot going on here. And so on the left panel, we've got just our synthetic nitrogen rate. We've got nitrogen leaching on the y-axis, and then the synthetic nitrogen rate on the x-axis. And what you'll notice, right, these black symbols are the individual lysimeters. So a lot of noise, as I highlighted, right? And we sort of expect this, right? We know there are concentrated flow paths in soil that might be picking up more of that nitrate as it's moving down. You might be picking up chunks of organic matter, potentially chunks of manure, right? If we don't have complete uniform application, we've got legacies of prior management. But on the whole, right, we see sort of what we expect, this increasing relationship between nitrate leaching in both the synthetic case and the synthetic plus manure on the right. So this turns out to be statistically significant. We're still playing around with different kinds of models. I think the slope is sort of interesting. If you look both of these experiments, on average, the slope is about zero point 0.3 when we look at the relationship between N rate and nitrate leaching. So what does this mean? On average, it suggests that about 30% of this applied N is lost to leaching when we look at the experiment as a whole. And that's, approximately about right when we look at other kinds of studies. Although there is, right, eyeballing the data, as we start bumping up that synthetic nitrogen rate, we do have some of this evidence for nonlinearity, right? This makes a lot of sense. When we really tap out on the crop demand, there's going to be more opportunities for nitrate leaching from these hotspots, especially when we've added some manure nitrogen on top. And
Stephen,
so,
I'm just going to interrupt.
yeah,
We did get another comment in the chat here, and if it's easier to answer this at the end, you can elect to do that.
what the heck? Let's take it now.
The statement is analyzing the groundwater is not useful, question mark, in the same place trial. I'm not sure.
Yeah, so that's a great clarifying question. So we're not analyzing the groundwater, right? We're analyzing the resin lysimeter that is installed at about just a little bit above 3 feet. So the goal is to put this below the root zone, but above the groundwater, right? So we're catching the nitrate that's moving through that soil profile through the lysimeter. That's great clarifying
That's
question.
what I suspected. I'm sure the asker will be appreciative. Keep going.
All right. Keep rolling here. Okay. So yeah, so on average, right, we see a bump in this manure that about corresponds with the amount of manure nitrogen that was applied. Okay. So I thought just, since I've got you here, I would briefly fit in another one of our trials where we're using these resin lysimeters just to give you some more insights about nitrogen leaching. And some of you may be familiar with WICST. This is a long-term trial down at Arlington where we've got a whole spread of cropping systems ranging from continuous corn to pasture, perennial pasture with grazing heifers. So a student of mine, Hariom Yadav, installed about 300 of these. And I think that, the replication was really worth the effort. These were installed during the same time period as this NOPP project. And I bring this up because we also have some systems that are totally reliant on synthetic and then this corn alfalfa rotation where we're applying substantial amounts of liquid dairy manure. And just briefly to give you a snapshot of the results, in this system, overall, we have pretty high nitrate leaching. Our corn treatments are maxing out about 80 to 90 pounds. So both the continuous corn and the corn after alfalfa, we see a bit lower during the soybean phase of the corn soybean rotation, and particularly in our second year of our alfalfa rotation. The first and third phases are a little bit leaky, we think, with just how and when the manure is applied with respect to termination of the crop. On average, actually, the pasture had surprisingly low nitrate leaching. We think one of the keys here is the presence of living roots year round. So despite the fact that there's a fair amount of nitrogen cycling happening with the animal excretion and mineralization, these systems are better able to hang on. And so I think this one of the take homes is really sort of the importance of this as close as we can get to continuous living cover to try to hang on to that nitrogen that's moving around our plant soil systems. Okay, so just to recap what we found from this Chippewa NOPP trial and the other experiment I just mentioned. So, we have some good evidence, right, of this increase in nitrate leaching with synthetic nitrogen application rate, tending toward this non-linear increase as we bump that nitrogen rate higher. As near as we can tell, it seems like the manure nitrogen and the synthetic fertilizer were similarly susceptible to leaching. And then, some evidence that these systems with continuous living cover can help us put a dent into the nitrate leaching. And so that really begs the question, well, how do we make this practical and feasible for you all to integrate, cover crops, et cetera, with your systems? Next steps, right? We're going to have more data accumulating over the years, so stay tuned for some more measurements from the Chippewa site and from WICST. We're still trying to dial in what these resin lysimeters are actually telling us and comparing them with other methods. And I want to highlight there's a group of us that are working on an extension pub really focusing on nitrate leaching and how we can measure and understand this coming soon from the Ag Water program. So just stay tuned for that. And like to particularly acknowledge Jerry for coordinating all of that lysimeter work at the Chippewa site. That was a big lift to get this done. The many volunteers who helped out and all of the lab work that went into analyzing those samples. So I will stop here.